Hello, my name is Jim Wilson, and I'm from the University of Pennsylvania, where I run the gene therapy program. And I'm delighted to be here at the 2020 FAST Summit in Gala. I've been asked to give an overview of current challenges to viral-mediated gene therapy, which I look forward to doing. Gene therapy is a new approach to treating monogenic diseases, many of which are rare. And developing approaches for the use of gene therapy in inherited neurologic disorders is a top priority for many of us. One of the reasons is the significant unmet need of this type of neurologic disease, in which there are over 3,000 genetic conditions that affect the neurologic system, with 75 affecting children. We'll start with an overview of what it means to go from bench to bedside, or a term called translational research. One begins with experiments in an animal model of the disease. Frequently, it's a mouse model, in which the goal is to show that your intervention corrects the disease. And you can also evaluate in the context of gene therapy whether overexpression of your gene causes any safety issues. Indicated here in scale is the size of the target organ, which is a mouse brain. Once having established the potential for benefit or efficacy in the mouse model, one needs to move to larger animals to demonstrate that you can scale the dose. Can we achieve the same level of gene transfer in a larger animal with a larger brain than we could in a mouse? But importantly, also to evaluate for any potential side effects to address safety concerns. And if you pass this gate of efficacy in the small animal model, and safety in the large animal model, you can move into first in human studies, where again, the primary goal is to show safety, but in some circumstances, it would also be possible to show some level of efficacy, whether it is clinical or the demonstration of a correction of a biomarker. Again, from when scaling from macaques to humans, there also is an increase and a higher bar because of the larger target organ, which is the brain. So really the crux of this problem is delivering enough of the gene to the central nervous system. The primary barrier to this is called the blood-brain barrier. So injecting our gene delivery vehicle, which was called a vector based on a virus called AAV, into the blood is one way in which we can achieve that. However, the vector does not transport out of the blood into the other side of the blood-brain barrier, where the neurons and the target cells reside. And this is because there is a barrier for transport of anything in the blood of any size, such as a vector, to the other side. And there are various ways in which we can improve this delivery that I'd like to describe. One is to develop a gene delivery vehicle, an AAV, that has the properties of being transported out of the blood into the brain. This has been achieved in mice using a modified version of a vector called AV9 that was discovered in my lab. And this just shows the remarkable results developed by a group in Caltech, transferring either the parent AV9 vector into the blood to target the brain, or a version that has been modified that can get across the blood-brain barrier. And this is a mouse brain showing the expression of the transgene really diffusely throughout the brain. Unfortunately, this same variant does not work in non-human primates. So it's a good tool in mice, but not an effective therapeutic for humans. probably one of the most important ways to improve delivery to the brain is the route of administration. So we just talked about an intravenous delivery to target the brain, which so far has not been achieved in larger animals. The other approach is to directly inject the vector into parts of the brain using a rather invasive approach where there's a burr hole created in the skull and a needle is inserted into the brain. This is limited both because of the safety concerns, but also the gene transfer is really restricted to where the needle is inserted. More recently, we and others have attempted to deliver vector to the brain by injecting it into the cerebral spinal fluid that bathes the spinal cord and the brain, either by an entry point in the lumbar space in something that's called a lumbar puncture, a common procedure in medicine, or injecting the vector in the same area, which is called the subarachnoid space, closer to the brain, which is the ventricles, or something called the cisterna magna. We will be discussing more what can be achieved using the best delivery vehicle and the most optimal route of administration. But even under the best of circumstances, the efficiency with which we can transfer genes in large animals is somewhat limited. And it has led to a focus on a subset of CNS diseases in which we can achieve sufficient gene transfer in large animals and humans. I'll just summarize some of these as a foundation for moving forward in Angelman syndrome. One approach is to inject a needle into a particular area of the brain to treat that localized area if that is all that you need, such as in Parkinson's disease. And that is indeed possible, as shown here. The other is to treat neuropathic genetic diseases of the brain in which there is the potential for cross-correction. As shown here, gene correction of one cell could lead to the production of a protein that could be transported out of the cell and taken up by an adjacent cell that doesn't have the corrective gene, improving both cells. And we and others have shown in many models that even low levels of gene transfer, 1 to 10%, can achieve a correction in many of the cells. That's not the case for Angelman syndrome. And then finally, we've shown that through these routes of administration into the ventricle or the cisterna magna, that we can target very efficiently the neurons of the spinal cord, the sensory and motor neurons, that could lead to correction of diseases in which only the sensory and motor neurons of the cord are affected. Again, not Angelman syndrome. So where do we sit with respect to the development of these treatments in biopharmaceutical settings and applications to patients? There actually has been a tremendous amount of progress made. So just my last count is that there are 21 clinical trials of AAV gene therapy in humans, representing 15 of the 21 coming from those storage diseases where there's a bystander effect, 5 out of 21 neurodegeneration, and then 1 a sensory motor neuropathy. But the good news is there's an avalanche of preclinical programs, those in animal models, that will be evolving into the clinical trials. And again, we don't have great numbers because much of this data is confidential, but we estimate about 122 named programs in biopharmaceutical companies. Again, the primary and the most common are those affecting storage and bystander effect diseases. There's also neurodegeneration, but importantly, there's an emerging new group of neurodevelopmental diseases such as AS, for which there are clinical programs emerging. And we expect this then to evolve into the clinic in due course. Just more details on what is in the clinic. Again, 21 total programs. The most common route of administration is into the cisterna magna, something that we've popularized, or directly injecting into the CNS or the brain tissue. In terms of the vector that's used, the AAV, the most common and the majority is something based on AV9, which is a vector we discovered at the turn of the century with a limited number with other capsids. So the way that we've progressed in our clinical trials of storage diseases and also sensory motor neuropathies, or what we plan to do, is directly injecting the vector into the cerebrospinal fluid, either the cisterna magna or lumbar puncture. And when we did a comparative study in monkeys, as shown here, it demonstrated that the most effective way of getting genes into the brain is when you inject the vector into this fluid closer to the brain rather than down further along the spine. These are data showing the amount of vector that is present in various tissues, brain, spinal cord, and then also non-CNS tissues when injected through the cisterna magna or lumbar puncture. And this is the total number of vector genomes per cell. And what we see is that the cisterna magna approach gives you much higher levels of gene transfer rather than the lumbar approach. So we've tried to determine what limits the distribution of vector when administered in the cisterna magna, and is it sufficient for some of these diseases? Well, if you review the anatomy of the brain with respect to its the cerebrospinal fluid compartments and vessels, what you see here in the schematic of a brain is the vascular distribution, in which you have these penetrating arteries that go from the surface into the deeper structures of the brain. And it turns out that the cerebrospinal fluid that bathes the brain in the cord actually tracks along these penetrating vessels, as shown here, that this is the subarachnoid space where the cerebrospinal fluid is, and it can penetrate right around these vessels in something called the perivascular spaces. So that does give some level of penetration into the brain. And when you do the experiment in monkeys where you inject a vector expressing green fluorescent protein into the subarachnoid space, and then analyze the brain tissue for the distribution of gene transfer here, you get a lot of gene transfer on the surface, which is where the vector would access the brain easiest, but you get these foci of gene transfer deeper in the brain, which we believe is due to the distribution of vector along these perivascular spaces. But with that said, even at the best of circumstances, we're looking at around 1, maybe 10% of cells that take up and express the gene. We believe that a longer range goal, and it may hopefully be shorter, to achieve better distribution of vector around the brain is using an intravenous approach. And one of the reasons is that the blood vessels of the brain distribute to smaller capillaries that are widely distributed throughout the brain, as opposed to those penetrating arteries for which the perivascular space or the cerebral spinal fluid is restricted. But the capillaries are really only 1 or 2 cells away from any neuron. And as I mentioned before, it is possible to identify an AAV that in mice, when delivered into the blood, broadly distributes gene transfer into the brain. So at least we have proof of concept. Unfortunately, we have yet to achieve this in primates, although we are all working on it very hard. It is possible to achieve some level of gene transfer into a primate brain with AAV9 as long as you inject very high doses of AAV9. And this is starting to look interesting, but it's limited by the fact that these high doses are associated with toxicity in primates, and the same dose of AAV9 or similar capsids in humans for treating other diseases, such as muscular diseases, have been associated with severe toxicity and even death. So we don't believe an intravascular approach with AAV9 is the way to go. But we and others are trying to develop variants of AAV9 that could potentially achieve the same result as this vector did in mice. We do this by creating populations of these capsids with different amino acid sequences, identifying hopefully one that binds to a receptor that would affect this transport and distribution into the brain. These are screened then in monkeys, and they're evaluated using something called a scorecard using next generation genome DNA sequencing. And this is just a preliminary review of one of these experiments where we've evaluated the monkey tissue for those capsids that deliver increased quantities of vector versus the gold standard, which is AAV9 or this variant PHVB, which in primates doesn't work very well. But at least we have some candidates that look attractive that we're further pursuing. So where do we sit with respect to the path to the clinic for gene therapy for Angelman syndrome? I'm just going to very briefly describe work done in my lab to try to demonstrate that AAV vectors can correct the disease, to determine how well this scales into primates with respect to safety and efficacy, and then conclude. So the gene we need to transfer is UBE3A, and there are many different forms of this, or at least several different forms: isoform 1, isoform 2, and isoform 3. We focused on isoform 1 and 2, driving expression of this gene and its protein from a promoter that is specific to neurons. And these studies have been performed in a mouse model of AS. So in this mouse model, we injected into the animals isoform 1 or isoform 2. And we've studied the expression of the transgene-derived UBE3A in the mouse models. And I just want to focus on these histological sections, which shows that indeed in mice, as you probably could expect because the brain is so small, that we can achieve pretty good gene transfer when injected into various routes, including right into the ventricles. So this allows us to ask the question, will this gene correction help the disease that these animals have? And so what we've done is studied the impact of vectors expressing either isoform 1 or isoform 2 into the ventricles of mouse models of Angelman syndrome. And we've studied the behavioral and neurologic abnormalities that are associated with this disease, such as longer stride length, hypoactivity, impaired nest building, and impaired motor coordination. Whenever we showed an improvement over the control, we have a green box, and whenever it was worsened, we have a red box. So far our data seem to suggest that isoform 1 is showing impressive correction of some of the abnormalities, and that isoform 2 not so much. So now the leap from mice to monkeys, using the same vector injected this time into the cisterna magna. And this is an example of monkeys in which we then analyze the brain tissue after injecting the vector for the vector genome. And here we have different parts of the brain, and then we have different parts of the spinal cord. Spinal cord is always targeted well. There are parts of the brain in which we get a fair amount of abundance of the vector genome. Now, when we analyze the tissue for RNA expression derived from the vector, it's present, But when we further refine the question as to how many cells are actually expressing the gene, it's a very limited amount. And at this point, we think it's probably insufficient to show correction if this translated to humans. The next question, though, is AAV gene therapy safe, expressing UBE3A? And here's an experiment where we evaluated the most important toxicity that we and others have seen using AAV into the brain of large animals, and with the concern that this may be present in humans. And this is actually toxicity of the dorsal root ganglia, which are the sensory neurons, which we've seen literally in virtually any model that we've studied in primates. The good news is that our vector so far, which is actually able to transfer genes to these dorsal root ganglia, has been about as safe as anything that we've seen, and no evidence of toxicity so far. So to conclude, where do we sit with respect to gene therapy for Angelman syndrome? What we've shown so far is that we can correct many of the abnormalities, neurologic abnormalities in the mice. And also, we show some level of gene transfer in monkeys that so far has been safe. But the crux so far is we don't believe we're at the efficiency of gene transfer in the brain that we need to be, despite the fact that so far there hasn't been much to worry us about safety. So the focus of the research is really about improving efficiency. And when we exceed our certain threshold, then we'll very rapidly move into the clinic. So again, I want to thank you for the invitation to participate yet again in this event, and this virtual event. And I hope this update has been useful to the families and the other stakeholders for AS. Thank you very much.